So I have a really long title. This is because Kathy emailed me at the start of the year, and I thought, well, I'll just throw everything in there and see what actually got covered during my talk. So this is work that has been ongoing in my lab, and I actually took a look at my rough cut from 2 years ago, and it actually had almost no data. It was just presenting what we're about to do. So I'm happy to report that I have some data to share with you today. Just to acknowledge some collaborators and students from both Montana State as well as University of Montana. So I also thought for a little time, I wanted to talk a little bit about a new project that's about to start right now. And this is moving on to tropical montane cloud forest. And only 5 minutes, but it's a new project that I wouldn't mind getting some input from people. Okay, so what I want to do is just start talking about some of the major themes of research in my lab. And the first one is this idea that the topography is really going to influence plant productivity. Now, this idea in itself is not particularly novel. We all know that as you go along elevational gradient, you're going to have differences in temperature and precipitation, and these are really going to drive the species composition in a particular area. But even within an elevation, you can think of the landscape position, so things like aspect, the landscape position. So in areas of convergent areas or divergent areas, that particular plant species growing in that environment is going to experience the climate really differently. So different insolation, different soil moisture, different temperature, VPD. Also things like subsurface flow initiation. So how connected are the convergent areas from the upslope areas? But my perspective is that, well, the plant physiology really matters. So I apologize. There's some issue with the MAC PC conversion. Some species can live along a very large, broad elevational gradient. They can survive across a very wide range of environmental gradients. And some species are very isolated to just one particular area. So different species have differences in rooting depth, photosynthetic rates, water use efficiency, how they build their structure, differences in sapwood area and leaf area. So while this idea that topography influences where species grow is not particularly novel, this idea of trying to integrate all of this together, I think, is. And this idea of trying to understand mechanistically how this all works, and then not just applying it for one particular watershed, but moving into different ecosystems. And how do these processes, can we scale from a plot to a landscape to an ecosystem, et cetera. So here's just an example of our field site in western Montana. This is a view looking out at one of our watersheds. The trees in yellow are larch. Western larch is deciduous, one of the few conifers that lose their leaves. And so this is in the fall. And Rob, I think you were with us when we took this picture in the fall. You can see that the larch are in these areas that are north-facing. Okay, so north-facing slopes. They tend to grow in cool and wet environments. Now, at the forefront, we have a ponderosa pine, and we have a Douglas fir, and these tend to grow in more exposed areas, so drier climates. And so this just gives you sort of a preview of what I'll be talking about later on in this talk. Now, the second theme of research in my lab is plant-water relations. And I don't think I have to convince any of you here that water is really important for plants. It's important at the leaf level, but also at the whole plant and ecosystem level. So here's a study by Sala in a grassland showing that as you increase precipitation, you're going to increase production, right? So you can scale this globally. If you look at evapotranspiration and net primary productivity, so we know that tropical rainforest has more productivity than a semi-arid grassland ecosystem. Now, while this is, I think, very important, I would argue that if you want a mechanistic understanding of this, it's not just total precipitation, right? What actually really is important for me to understand is the form and frequency of precipitation over time. Okay, so what I mean by this is I'm going to give you some examples. So for example, in the Sierra Nevadas, we know that snowpack is a more important water source than rain. Here's a study by Trujillo comparing two different water years. So this is a dry year, 1990, versus a wet water year, 1998. And the green is the NDVI, which is used as a proxy for productivity. And what you see is that in the very wet water year, you have these large circles, which tell us these are the peak snow water equivalent, so snowpack. So high productivity year, large snowpack year. low productivity year, low snowpack year. So snowpack is very important in this ecosystem. But we also know that conifers don't depend entirely on snow, that they can take advantage of rain when it is available. This is a study looking at ponderosa pine, 3 different populations, Arizona, California, and Oregon. And here's the Arizona population that receives quite a bit of monsoon rains. Okay, and so if we look at the isotopes, which I'm not going to go into now, I'll go into a little bit later. If you look at the isotope of the cellulose of the population over here in Arizona, you see that they actually incorporate a lot of this monsoon rain into their rain signal. Okay, so when this rain is available, this population that sees monsoon rain is going to incorporate that. Now, it's not just water from the roots that are particularly important. Some species, water from the atmosphere can be very important. Redwood trees is a great example. They live in the coast of California, Oregon, where in the summertime they see a lot of fog. So here's a study over 3 years. In these periods right here, this is the summer period where there's significant fog cover. And here is on the y-axis is the proportion of xylem water as fog. So this is, again, using isotopes. And you can see during the fog season, all of these different plant species, a significant portion of their xylem water is fog. Now, what's particularly interesting is, so here's redwood, Sequoia sempervirens, it's in the black squares. But these two right here, these are actually understory plants that don't actually intercept any fog. But what they rely on is fog that's intercepted by the canopy of the redwood tree. It condenses, it drips, and it condenses and it becomes fog drip, and then these plants take it up through the roots. So fog drip is really important for these understory plants, and a place in the summertime where it receives absolutely no rain. This can also be the case in tropical montane cloud forest. This is a study looking at this particular plant that has different life stages as it grows. So when it's growing as a juvenile, it's an epiphyte, so it grows solely in the canopy. And you can see that the xylem water right here matches almost exactly with the range of values from cloud water. So it's receiving all its water from the canopy. And as it grows and it puts roots down, right here, when it becomes an adult, now its roots are tapping into the soil, and now the xylem water reflects soil water. So different species can also change their source of water over their lifetime. So to really just summarize these broad research questions in my lab, the first is looking at landscape position. And then the second one is looking at how different types of precipitation drive plant water use and productivity. So what I'm going to do is talk mainly about the work that we're doing at Lubric, and then if you have time, talk more about this cloud forest study. Okay, so this is the Lubric experimental forest. This is in western Montana right here. It's operated by the University of Montana School of Forestry. These are the dominant tree species we have there. Douglas fir is highlighted in green because that's the one species that grows along the entire elevational gradient. This is a watershed that we are working in. These areas highlighted in red are sites that we've established and heavily instrumented. So we have 2 sites that are low elevation, 2 sites that are high elevation, 2 sites that are south facing, and 2 sites that are north facing. And the goal of this was sort of to establish sites that had a very wide range of climatic differences. Now, before I started working at Lubric, my collaborator, Kelsey Genso, at University of Montana, he and his graduate student, Zach, had gone out and cored a lot of trees within this watershed. And what they were looking at is how does topography influence productivity at an interannual time scale. So they were coring trees, looking at the incremental changes in the basal area increment over time. And so what you see is that the wetness index, so the more wet the environment, the more the plants grow. Okay, so again, this is not particularly that novel, but this is just to confirm something that we were trying to test. But this is at the interannual time scale, and from a plant ecophysiologist perspective, what I wanted to know is what happens at the intraannual. Okay, so within a particular season, how does landscape modulate water use and productivity? And then how do different sources of water tie into this? And more recently, now we're looking at how this landscape controls nitrogen dynamics, so nitrogen availability and also nitrogen uptake. And this is sort of the very new stuff that we're still trying to make sense of the data. In each of our sites, so our 4 sites, we have met stations, so we're measuring air temperature, humidity, collecting rain. We have soil moisture at 3 depths, both in the hollow areas as well as the side slopes. We also have wells at our site, so we can measure subsurface flow. We have transpiration measurements using sat flow sensors right here, and we have growth measurements in dendrometers. Okay, so I'm sort of just going to go through those 3 questions that I just posed. We also collected isotope samples. We collected rain, stream, and snow. These are our potential sources of water. And then we collected plant and xylem and soil samples about every 2 weeks. These samples come into our lab. If you've ever been in my lab, this is the contraption that we extract the water from. And then we analyze it in-house on the Picaro water isotope analyzer. We have undergrads working around the clock on the extraction line. So if you ever hear a really loud hiss coming out of my lab, it's the liquid nitrogen that's going off. Okay, just to show you some site differences, these are just annual sums. I just wanted to highlight very quickly that we do see differences between our different sites. This is our low elevation south aspect, our driest site by far. Here's soil moisture in the hollow. It's higher than soil moisture on the side slope. This graph right here, this is depth below ground. This is telling us the connectivity between upslope and downslope. So these areas that are shaded tells us that there's water actively moving below. So there's connectivity in subsurface. Here's our high elevation site. Okay, so again, our hollow soil moisture is higher than our side slope. But if you look at this connectivity now, this region shaded by gray, it's a lot more connected. Okay, so the hollow areas are receiving a lot more. It's receiving a lot more soil moisture coming up from upslope, but flowing laterally downslope. Okay, so this first question, looking at topographic influences on interannual tree productivity, this is work spearheaded by my graduate student Justin. These are the dendrometers that he has on the trees. These are pressure point dendrometers, so he has 2 on either side of the tree. This is drilled into the tree, and this is just a little pressure transducer. And so when the tree swells, it moves out. When it shrinks, it moves in, and it's just recording this every 30 minutes. Now, one of the things we're trying to understand, okay, so this is an intra-annual time scale. So what's happening here with growth? So here is a high elevation and low elevation site. Here's the soil moisture for the high elevation, and here's the soil moisture for the low elevation. Now, when we think of water stress on trees, a lot of times you might think of soil moisture being the most important factor, right? The soils are dry, the tree is going to decrease transpiration rates, decrease growth rates. But this is just the supply part of the story. There's a demand component. That's the vapor pressure deficit. So water is not going to move out of the tree unless there's a really high difference in vapor pressure between the outside and the inside. Right? So the tree is always saturated. So if it's very dry on the outside, water is going to be pulled out. If it's high relative humidity, like in a foggy situation, water is not going to be pulled out as much. Okay, so right here, this vapor pressure deficit, this is really the demand. And so Justin's come up with this really cool metric that incorporates both the supply and the demand, and it's this gray line. So this is 1 over VPD, 1 over volumetric water content, which is soil water content, times VPD. Okay? So what this tells us is that high elevation, the supply and demand is not really that high. There's not that much of a draw. But in the lower elevation, okay, right here as you go into July, there's a pretty high demand. Soil moisture is pretty constant. But this is really the VPD that's driving it. So it's really dry. Okay, the plant's going to experience a lot of water stress. So if you look at this change in the stem radius now, these are the dendrometer measurements. You look at trees growing in the hollow and the slope and the high elevation, this stem radius change is pretty much the same. But if you start looking at the lower elevations, the drier environment, higher VPD, you see that when you start coming into the July mid-season, midsummer season, it starts diverging. So the slope trees slow down in growth while the trees in the hollow continue. So what Justin's working on right now is a model that predicts growth using simply volumetric soil moisture and VPD across our entire landscape. Now, one thing that complicates this is that we don't actually know that the stem radius is growth, right? The tree is going to swell and shrink as it's taking up water and losing water. So what we want to do is we want to separate the growth curve from this curve. So the growth curve could be this blue line. It could be that growth matches perfectly with the swelling, but it could also look like this. And we don't know with just dendrometer data. So what we're working on are these things called microcourse. These are, they're microcourse, as the word describes. This is a tiny wood core that we get from this little instrument that you punch into the tree. You can take out a tiny, tiny wood core. And Justin's been doing this every single week. And the idea is that you can count the number of new cells that the tree has been putting on every single week. So we've been making a lot of slides, staining them, in the process of putting them under a microscope and now counting the number of cells. And so the idea is that we can put a growth curve to that stem radius curve and actually validate that curve and try to sort of tease apart what is growth and what is swelling. Now, another way we can get at this swelling is to look at transpiration. So in the same trees that we have dendrometers, we also have sap flow sensors. And the sap flow sensors tells us how much water is moving through the tree. We're hoping that can tell us something about the swelling and shrinking. And so here, just to highlight, trees growing in the hollow, trees growing in the slope, you can see that the hollow trees have much higher fluxes of water during the day. So slope, there's less, but you can also see that they both sort of experience something similar around day 260, where the transpiration rates decrease a lot. Okay, so if you add up all the daily fluxes, you can get a cumulative graph. So you see that trees growing in the hollow, they're using a lot more water than trees growing in the slope. So the idea is to use the transpiration data and then the microcore data to help us separate the growth from the swelling and shrinking. Okay, so we know that there's these differences in growth patterns of trees growing in different landscape positions. But now we want to know, do different sources of water influence some of this? Okay, so are they using snow at the high elevation because it receives more snow? Are trees in the hollow using deeper water? Are they using water that's subsidized from upslope? So these are the kind of questions we're asking. And to do this, we're using stable isotopes of oxygen and hydrogen. This is just a mirrored water line, so this is the relationship between oxygen and hydrogen. Most of the points globally fall on this line due to different fractionation processes. You tend to get cold region values are more negative, warm region values being more positive. We also have values that are more negative tend to be more snow-like, higher elevation, deeper soil water. Values that fall in this range tend to be things like rain, lower elevation, and shallow soil water. So because of these differences in the isotope value of oxygen and deuterium, we can use this to sort of track water source use in trees. So the first thing we did was we analyzed the water samples that we collected at our site, so rain, stream, and snow. And these values fall where we expect them to fall. The snow value is a lot more depleted, a lot more enriched. There's a huge spread in the rain, and this is probably because these are more spring-like, spring snow fleet values here, and these are probably midsummer, late summer values. And our stream values, you can see, doesn't change very much throughout the season. This is probably groundwater that's contributing to the stream flow. Now, we also looked at the isotope values of precipitation elevations. Okay, so here our rain samples are in circle, and this is just our one snow value. We went out at peak snow pack, went out and took cores along transects and analyzed it. But one thing you see is that there is this elevational difference in the isotope ratio. So the values that the precipitation falling at higher elevation tends to be more negative consistently than the precipitation that's falling at lower elevation. So this sort of set up our next question, which is, okay, so if the values are more negative at high elevation, which is telling us it's probably more snow, colder, then is the value in the trees reflecting this? Are trees at higher elevation using more of a snow melt-like signature, while trees at the lower elevation are using more of a rain-like signature? So to further help us resolve this, we also analyzed a lot of soil profile data. So what we did was we dug a soil pit down to 40 centimeters and collected at different depths. Okay, and then we did one profile in the slope and one profile in the hollow. These are our 4 sites, and these are our 4 sites through time. This is a very busy figure, so there's only 2 main points I want you to take away from this. These are the rain values collected around the same time we collected the soil samples. So the first is that if you look at the upper soils, so upper 10 centimeters, the upper soils really reflect the rain, right? So these upper soils track the rain quite well. But the lower soils, so below 30 centimeters, they don't change very much through time. So if you go from date to date, they remain pretty stable. The second sort of takeaway from this is that the hollow soils are more negative than the soils on the slope. So the blue lines, the light blue lines, tend to be to the left of the dark blue lines. Now there's some exceptions, but usually, typically, they're more negative. So soils in the hollow are more negative. They're probably deeper. More negative means more snow melt signature. So again, are the trees then in the hollows using more negative, or do they have more negative isotope values in their xylem water compared to trees growing on the side slope? Okay, so it's the first question we asked. Okay, so now this is the xylem water. This is water extracted from the trees that we collected. This is hollow versus slope across the entire watershed. And we do see this, yes. So trees that are growing in the hollow are more negative than trees growing on the slope is using probably deeper water or water with a more snow melt-like signature. But now we asked, what about trees at high elevation versus low elevation? Are high elevation trees using water with more of a snow melt-like signature? And across the watershed, there's really not that much of a difference. And this was sort of perplexing for us. We thought, well, if the input of water is different, then why does it seem like they're using the same water source? So what we did was we broke it down by species. So again, this is the xylem water now broken down by different species. And one thing we noticed is that larch in the cyan blue, they tap into a very deep water source, and they use it consistently all year long. They're not using any of the rain. So if rain falls, it would fall in this range, and you would see a shift. But larch doesn't use any of that. Larch is tapping into a very deep water source consistently. Now, other species like Douglas fir and subalpine fir, they're using more of this rain that's coming in. At low elevation, the reason why we didn't see this difference between high and low elevation is because ponderosa pine, to our surprise, is actually using very deep water. We had thought that the lower elevation, ponderosa pine would be using rain that was falling at our site. But the isotopes tell us that they're using very deep water. I mean, deeper water than some of these other species. And so when we thought about this a little bit more, it makes complete sense. You know, the lower elevation site where ponderosa grow, it's one of our driest sites. The soils get almost completely dry by July. And so you can imagine the upper fine roots during that time probably finesse. So even if rain falls, those fine roots probably aren't taking up any of that moisture. And so it's probably a safe strategy for ponderosa pine to just tap into a deep water source and use that deep water source all year long. The high elevation soils, they're moist for longer. When the rain falls, those fine roots are still active and probably taking up some of that rainwater, which is why these values up here are a lot more enriched. Now, Douglas fir is the only species that grows along our entire elevational gradient. And so we compared how Douglas fir were using water between the high and low elevation. And you can see that there's really no difference either. Douglas fir is tapping into almost the same depth of water as low elevation as it is in high elevation. So this is still, you know, the depth that they're using water is originally different how we had predicted, but you can explain it physiologically why they're tapping into these different water sources. So this is just showing you 1 year of data. We have 2 more years of data that we finally finished extracting, and our Picaro's back from the factory, so we were starting to analyze to try to strengthen this part of the story for two more years. Okay, so the next step, what I just showed you was water use within a year. What about historic water use? How long have these trees been relying on snowmelt and rain? How flexible are they in different water sources? Are there particular years where one, or particular decades when one water source was more abundant than the other? So next step is to start looking at wood cores. So the water that the trees take up is incorporated into cellulose. Okay, the cellulose gets laid down here, and so within a particular ring, you can analyze each ring, extract it into cellulose form, and then analyze oxygen isotopes to start partitioning water use per year, but going back over 100 years. And this is what we're trying to do next. And so my PhD student, Tim, will be working on this. Now, this sounds really simple, but it's not, because in addition to the source water, the environment also influences the oxygen isotope. So under a very dry environment, you have a lot of fractionation of the leaf water, which makes that leaf water isotope very different from the source water isotope. And so we have to account for differences in relative humidity over time. And so this is something that we're working on right now through both modeling and using SNOTEL data of temperature and relative humidity. Okay, the third question is now looking at landscape controls on topography, or landscape topography controls on nitrogen availability and uptake. And this is in collaboration with Dr. Yurko Yano and my new PhD student, Claire. This is the image of Yurko and Claire installing a lysimeter. We did this last summer, so there's no lysimeter data yet. And here are all the silt extracts that I'll just talk about in a second. So one of the things that we wanted to understand is, we know that topography influences productivity, but what about nitrogen availability? So, if you go hiking out, well, not hiking, maybe skiing out in the wintertime in a mountain ecosystem, you know that there are places where there's really deep snow, really shallow snow, right? There's a lot of heterogeneity in snowpack. So if you have a very deep snowpack, the soils underneath that are going to be very well insulated. So under well insulated soils in the wintertime, you might have a lot of mineralization happening. In an area that's more exposed, like on a slope where there's a shallower snowpack, the soils might get very cold, lower rates of N mineralization over the wintertime. Now you can imagine when spring comes and that snow melts, you can get a pulse of nitrogen moving through the system that might be very different if you're in a well-insulated area versus a shallow, non-well-insulated area. So to get at this microtopography, what we did is within each of our 4 sites, we set up a series of transects. So we're going up the watershed right here, and we've got these transects going, 5 going across. So we have 2 at the very top, 2 in the middle, and 1 sort of in the hollow or the gully. We've done this 5 times. At every one of these black diamonds, we took a soil core down to 15 centimeters. We extracted the nitrogen using KCl extraction, and we did this every single month last year. Plucked the roots, all that stuff. It's a lot of work. Here is our data. So we're still trying to make sense of this data a little bit. Again, a very busy figure, but I'll walk you through it. The colors here correspond to the different sites. So green and blue are high elevation sites, north and south aspect right here. The circles are hollow, and the triangles are slope samples. So if we just look at the ammonium data right here, one thing that stands out is you can see that all the triangle data, so all the slope, are pretty much down here, very low. Some of the highest ammonium rates are from this north aspect high elevation, our coolest site, our wettest site. So we have this flush of ammonium at the start of the season. So this is when the snowpack is still actually on the ground. It's melting, but it's still present. And then it decreases. And then we have this really large increase in ammonium. And this is around the time that we have a series of rain events that have come in. And so we think what's happening is that we're getting this buildup of ammonium because mineralization is actually happening. But something is preventing it from going into nitrate. So the nitrification step is not happening because if you look at nitrate, nitrate is decreasing. I'll get to this point in just a sec. So we see the biggest pulse of nitrate available at the start of the season when the snowpack melts. right? And then nitrate continues to decrease over time. And it seems like there's some sort of bottleneck where mineralization is happening, but nitrification isn't. This right here, this particular site, our high elevation North Aspect site, that site has alders, which have nitrogen fixing bacteria, and so you might explain why that one is so high. So, okay, so we've got this pattern of nitrogen availability. We're going to repeat this again in the summer. We also have resin strip data, so going back to this. We have weather resin strips that's looking at exchangeable nitrogen in these plots right here. We also have lysimeters now in these, and so we're going to have lysimeter water that we'll analyze for nitrogen. We have resin strip nitrogen data, and then we'll have soil extractable nitrogen data. So now the next step is looking at nitrogen uptake now. Okay, so we know these patterns of availability, but are plants in sync with this availability? Are they taking up nitrogen in the same way that it's made available? I think most people, including me, when I took ecosystem ecology, we learned that most temperate ecosystems are nitrogen limited, right? The idea that if you add more nitrogen, they're going to grow more. But there's this growing body of literature that suggests conifers, evergreens, are really not that nitrogen limited. There's a really good evidence that they store a lot of nitrogen in an inactive form of residuo, or they store it as arginine. So if you look at how much nitrogen is actually in the leaf, and you predict what photosynthesis should be, it's actually completely off. And so what we're thinking is, okay, so there's nitrogen available at the start of the growing season, but what if the plants aren't taking this up? There's also a lot of evidence that trees, when they grow their new needles, are using nitrogen from the year before. Okay, so they're using stored nitrogen. So there's this big pulse of nitrogen that's available at the start of the season. What if the trees aren't taking it up and they're just using old nitrogen? Not using this new nitrogen, then what's happening to this new nitrogen? They're taking it up and storing it. How is this nitrogen getting translocated? And so Claire is going to be working on this, where she is actually going to be looking at nitrogen uptake across the season. So the first experiment involves actually measuring nitrogen uptake via the roots. So she has quite a few seedlings in the greenhouse. What you do is you put the roots in a solution of a known concentration of nitrogen ammonium. Over time, you watch how much of that nitrogen is taken up, and then you can clip the roots, see how much nitrogen went into the roots, and calculate flux rate that way. The idea is to also go into the field and find some small trees that we can dig up the roots and do an in situ uptake experiment there as well. To sort of get at this translocation issue, Yuriko and I have talked about doing sort of a mass balance approach, and so we would find smaller trees, and we would be collecting nitrogen or collecting different parts of the tree. So buds, new needles, last year needles, this year's branches, last year branches, wood, and basically measuring the nitrogen content. If you know the mass, we can do a mass balance for the entire tree. We would do this at 2-week intervals throughout the year, trying to get at translocation. So hopefully combining these two together. Okay. Take a drink of water. That was a lot of talking. Are y'all with me still? This part is relatively short. Okay. So I'm moving into another type of forest. These are tropical montane cloud forests now. I don't even know how I got into this, except just some conversation that I happened to have and started talking more and more about it and really got interested in this topic. And really just one of the reasons is because this uptake of water by leaves really intrigues me. I think it's something that is completely overlooked in a lot of ecosystems. You know, I think most people still learn that the most direct way for water to get into a plant is via the roots, and there's a lot of ecosystems where showing that uptake by the leaves is actually quite important. It could be direct uptake, or it could be condensing water on the leaves, which then drips and then taken up by the roots. But you know, the atmospheric moisture can be a very significant part of the plant's water budget. And so this really got me interested in this. And more importantly, you know, these tropical montane cloud forests, they're disappearing very quickly. So they represent a very small fragment of all tropical forests. I think there are 3%. So of all the tropical forests out there, only 3% are tropical montane cloud forests. They sit at the very tip top of these ecosystems. I'll point out right here. You see the Galapagos Islands, and there's cloud forest there. Come back to that. So why are they important? And why do we care? And what are tropical montane cloud forests? Well, the definition is very loose. If you look it up, it'll say it's frequently immersed in clouds. But this cloud is a really important water source, particularly during the dry season. And so in the tropics, you have a wet season and a dry season. During the dry season, this is a very important water source because you're not getting any rain, but these trees and other vegetation, they're essentially stripping moisture out of the atmosphere by having it condense, and it's dripping into the ground and it's providing water to understory and even downstream communities. They're usually covered in bryophytes or epiphytes. The trees in the tropical montane cloud forests are smaller. They're shorter in stature compared to lowland tropical rainforests. If you go up there, I think it's about 85% of the vegetation or of the trees are covered in some sort of epiphyte, bryophytes. These TMCF forests are also hotspots of biological diversity, a lot of endemic plant and animal species. And what I think is particularly interesting is that these are sort of the headwater ecosystem of tropical ecosystems, right? These are the very top of the mountains that's removing water from the atmosphere and providing water to downstream communities. And again, very important during the dry season. So there's a lot of potential connectivity between the tropical montane cloud forest and tropical lowland forest that still remains to be explored. There is this hypothesis put forth about 10 years ago, and it's gained a lot of interest, but there's been very little empirical evidence. And there's this idea that as climate changes and it warms, the height at which the clouds form is going to increase, right? So here's your zero isotherm right here, and as temperatures increase and warm air moves up, it's going to push these clouds up. So now these plants that used to live in the cloud forest are going to live in an environment where there are no clouds. Now these plants have adapted to live in a very wet, humid environment. They're very prone to drought. So the question is, if the clouds start moving up, these plants right here start experiencing drought. Once they start dying, there's potential feedbacks. There's also now no moisture coming in during the dry season because there are no clouds. There are no trees to strip that moisture out of the clouds. So if you go through the literature, you'll read a lot of studies that cite really two papers that model this, and there's almost been no empirical evidence to show that this is happening. And so my colleague Diego and I, we started talking about this, and we thought, gosh, I wonder if tree rings in the tropics could tell us something about this cloud lifting phenomenon. I wonder if we could go out and course in trees and look at this. One of the difficulties is if you go into the tropics and you look at a tree and you core a tree, there are almost no rings. And it's because rings form because you have early wood and late wood. And late wood in temperate ecosystems, they develop because the tree experiences drought. Okay, so in the tropics, it's wet all the time. They have, beautiful hardwoods that have no rings. That's why we like teak, right? But so it makes it really difficult to go out there and core a tree and try to use ring width as a way to reconstruct climate somehow. But luckily, we have isotopes to do this. So this is just a conceptual figure by a study by Kevin and Ecohydrology. And this idea is that as you go through a wood core, there should be some cycle that's reflected in the oxygen isotope signature. So if you have warm temperatures, reduced cloud precipitation, it's going to bring the isotope in this direction. And if you have higher rainfall, cooler temperatures, it's going to bring the isotope in this direction. And so in the tropics, if you have a wet and a dry season, as you go from the wet to the dry, you might see this seasonality in the isotope data, which can tell you something about the environmental conditions, but also give you sort of an age of the tree by counting the number of cycles it oscillates through. So they tested this in Costa Rica. You can see this is the bark of the tree. Now they're going into the cambium. What they did was they just sectioned the core at set intervals and analyzed the oxygen isotopes. So here are four different trees, and you see this nice cycle, right? And so you can sort of, I'm not a dendrochronologist, this is not the part I do, but you can put these together and create a chronology of the particular area. Now what I am interested in are these differences. Okay, so what is the difference between this value versus this value, and what's causing the difference here? So before I was talking about tree rings, I was saying that the cellulose incorporates the source water and the environment. So if you have a very dry environment, that can actually play a really large role in changing the cellulose isotope right here. Luckily, in the tropics where it's really wet and humid, because the humidity is so high and constant through time, it turns out that the largest signal from cellulose is actually the source water. So what this is probably telling us is that there's precipitation coming in the wintertime, in the summer, winter, summer, winter, summer. And so our idea is we could go out and core some trees. Maybe this could tell us something about what's happening long term. So here's that figure again. This is the mirrored water line. And like I was saying, snow tends to be here, rain tends to be here. Fog is actually very isotopically enriched. It's very distinct from rain. It's almost more enriched sometimes than ocean water, so you can have positive values. And it's just because of the way fog forms and the way it's removed from the atmosphere. So if we can use rain and snow as end members to partition water source, maybe we can use fog and rain as end members to partition input of water from fog and input of water from precipitation. So this is sort of the working model that we have right now. This is the idea that if we can go out and core trees that are not in the cloud, in the transition area, and in the cloud forest, this particular tree now, okay, if this particular tree is experiencing cloud lifting through time, the clouds are shifting up or through time, maybe in the past it has this value of about 30 per mil, showing us that it's relying a lot on fog. But over time, as that reliance on fog decreases, that value might change to something like 22, because fog is more enriched than rain. Now, if we then look at the oxygen isotopes, this is our prediction, is that in the past, when it was growing in an environment immersed in cloud, it's probably less water stress. So it has a very negative carbon isotope ratio. But as those clouds have moved up and it's experiencing more stress, these values have become closer to zero, so moving this direction. So this is sort of our working hypothesis that if we can detect changes in this cloud lifting, these are the predictions that we would see in the oxygen and the cellulose, the oxygen and the carbon isotope data of the cellulose that we collect. So in June, I'm going back to the Galapagos. This is the Galapagos I think most people think of being dry, scrubby, but it's actually quite foggy too. So this is at the top of one of the lakes. This is called the Gaurua. It comes through during the dry season. Sometimes it is so foggy up there you cannot see the person in front of you. You have to trust me when I say it's really foggy. And so the idea is we're going to go and core trees along an elevational gradient and analyze the oxygen isotope and the carbon isotope along these tree cores to try to detect any changes in cloud lifting, at least in the Galapagos. Okay, with that, thank you for coming, and questions? Jack.
I'm wondering about your comments about the nitrogen cycle. As you know, we've been working on nitrogen uptake in Conifer about an hour north of you the last couple of years. So the idea that conifers aren't nitrogen limited, I'm trying to reconcile that. It's like 100 years of research in Scandinavia find nitrogen that conifers and seeing huge growth. And then the other idea that if you take a system near steady state and you have litter fall fluxes and root turnover amounting to maybe 150 kilograms of nitrogen per hectare per year, if trees weren't taking that up, mineralization would proceed and losses would be way higher than you observe in many conifer forests. So trees have to be taking it up.
Oh no, I'm not saying they're not taking it up. So back to your comment about the Scandinavia, some of those studies in Scandinavia are the ones that show that they're also not nitrogen limited. Those are the studies that show that Norway spruce are accumulating arginine in excess of what you would predict them to need in order for, for maximum photosynthetic rates. So I think in this, of course, if you give a particular tree, nitrogen is going to grow. But in its natural environment, the idea is that maybe it doesn't need that much more to actually survive. I mean, these are long-growing plants that have evolved over millions of years to deal with really low nitrogen content. So if there's nitrogen that's available, they're probably taking it up, but they're not necessarily using that. So the uptake rates or the uptake patterns across the season might be decoupled from the availability, if that makes sense. So if you have peak nitrogen available at the start of the season and it drops down this way, it might be that trees aren't taking up the same way because they have this excess storage of nitrogen. So I'm not saying that they're not taking it up. I'm just saying that the timing of uptake might be very decoupled from the timing of availability.
Also a product of the uptake.
Yes.
Almost any mineralization assay doesn't include.
Right.
So I mean,
yeah.
If you look at the internal turnover, just like resorption, it's
so we're,
way too low to account for nitrogen demand.
Right. So that's why we're trying to do this mass balance with the whole tree, is to look at some of that resorption over the winter time. How much is taken, you know, because conifers have also very high nitrogen use efficiency, right? So if you look at whether that's looking at the litter nitrogen content versus what's in the canopy, or from a more gas exchange perspective, if you measure photosynthetic rates in a needle and you try to correlate that with nitrogen content, there's not a good, clear correlation between those two. So, I mean, one of the reasons why we wanted to look at this nitrogen uptake is that there are very few studies that are looking at seasonal uptake dynamics. And I know, you know, you and your student doing that, which Claire met and was trying to talk to you about this. We just don't know when they're taking it up, and it would just be really neat to know that, right?
I had a question about the cloud lifting story. So that's being driven by increased surface water temperature?
Or just air temperature.
Air temperature. Okay.
Yep.
Well,
and surface, yeah.
Right. So I wonder, is you mentioned that you're going to be looking to see whether things have been changing over time, but you would think that the dynamics that are affecting cloud height are very quick, and you know, there's not a lot of memory in that system. So, but presumably at these places we know what the surface water temperature is, we know what the air temperature was for quite a long time. These places have been studied for a long time.
Well,
so would it be? Are you thinking about looking kind of year to year to see what were the conditions like in this year, and then what did we see happen in that year?
So there's quite a bit of information when it comes to sea surface temperatures. You're right, but the cloud height base, yeah. there's almost no data. There are only 2 studies that we've known that have done this, and it's because these cloud ceilometers are really expensive to purchase, and so people usually go into airports. So, like, airports have great data on this. They're just not in the tropics, right? So in places that there's a lot of good met data are places that you can use these ceilometer data. But when you go to the tropics, there's very little long-term, any sort of met data. And so our idea is that we don't need to go back year to year because we're using the trees to tell us what's happening in the past. So this is the present, and this is the tree core going to the past. And so the question is, has there been a systematic change over the last 100 years in the source water that's coming in, and using that as a way to estimate if cloud frequency or height has changed.
My point is that the cloud height is going to be bouncing around.
Right. Yes.
And there may be a trend to that, but if we could somehow kind of back out what might have been going on in cloud height in a given year, based on sea surface temperature and what we know about air temperature and whatever. I mean, there are, I'm sure in your studies about a fair amount of weather data going back.
We don't.
I think the Galapagos would be fairly well studied for at least last few decades.
Yeah, I mean, they have them at the research stations, which are right by the ocean.
Right.
So nothing really high up. Yeah, I mean, so this is sort of integrating, each year it's integrating the entire year. Mm-hmm. So it could be cloud frequency. I mean, the idea is that if we can sample along elevation gradient, we might be able to detect where along this gradient that cloud has been shifting over time. Yeah.
Rubric, you showed that some trees take up rain, others take up snow, if they're watching snow. Given that, climate going forward, we're going to have lower snowpacks, more rain and elevation. Are people predicting major changes in the species composition of conifers?
That is a great question for someone like Ben. I'm not sure we've talked. I mean, that's the thing is I'm not sure how these preferential uptakes are, or I don't think they're incorporated into models. But you know, it's not that the trees are choosing one source over the other, right? It's sort of just what's available in the landscape. And so if more snow is available and lasts longer, that's what their signal is going to look like. But you're absolutely right. I mean, I had thought ponderosa pine would be taking up more rain because it lives in lower elevation. But we see that it's not because it's too dry, so it has to go deep, and then the trees at higher elevation are using more rain because the soils remain moist for longer. So it's sort of opposite of what I would predicted, 5 years ago. Yeah, but I don't know how.
Well, it's also interesting to think about this new index you have relating soil moisture and BPD, because right now kind of one emerging paradigm for tree mortality is based on BPD driving more potential and causing cavitation, hydraulic failure. And so it's also interesting to think about using the new kind of index that you presented as another way to understand the coupling of soil moisture versus BPD demanding and causing that mortality. So for the partitioning of the snow and the rainfall, it could be important if soil moisture is driving the water potentials, but if it's really a BPD issue, then maybe it's not important.
Yeah. Definitely.
Yeah, I was just curious about your isotope values with depth in the soils and how that aligned with the source water story. Because it seemed like your observations of rain are always higher. Maybe we could look at the figure, but your soil values were always lower. So I assume your rain observations were at a point of a season, and so the fact that those profile values are lower, would you explain that as a snow melt thing or as just
down here, you mean?
Rain? Yeah.
Yeah, I think what's happening down here is that the snow melts, and then even when it rains, I mean, I have this great picture that I didn't show, but there was this massive rain event, 2 days straight rain. And you go down and we dug a pit, and it went down 10 centimeters, and then it was just, you could see where it was dry. So it's not getting down super deep. So it's the upper soils that are really affected by the rain events. And then I think deeper down in the soils, it just don't change that much because rain's not getting down there. It takes the next snow melt to then change the entire profile potentially.
But so don't those values align with your tree values, low and high
Yes.
elevation? You know what I mean?
Yeah.
So you were talking about roots going deeply, but these are 40 centimeters.
Right, so below 40 we don't expect, it's hard for us to dig below 40, but other studies have shown that once you go below 40 centimeters, the profile doesn't change very much. And so the roots are probably getting it at 40 or deeper, probably going deeper in some of these cases, but we just don't have the values for that. Versus the shallower ones, the upper soil, the upper isotopes for some of the samples in the high elevation sites have relatively enriched, so it's less than -8 per mil. So they're using pretty shallow soil surface water in some of the higher elevation, like Douglas fir species.
So you're interpreting these lower values as snow melt in general. This is
Yes.
snow melt by soil.
Yep. Yep. So the snow melts, it sort of sets the profile, and then the upper 10 centimeters is going to change as the rain falls, and the rain isotopes are going to change at upper 10 soil centimeters.
So how would that align with the overall water balance? So I guess the way to test that idea would be to think about how much precip occurs as rain.
Yes.
And think about whether it,
yes.
whether it makes sense for you measure.
I think what complicates this is, so in the hollow, what we're not sure right now is that value there because it's just a lot of snow and over time it remains there, or is there actual subsurface flow coming from upslope that's subsidizing this, that because it's deep water it looks like snow melt. So we have wells this summer that Kelsey, we have those cap rods inside, so we can actually measure the movement of water and try to untangle that part.
Joe, I have a question about the isotope values in the plants, and I think it might just be one. No, it was versus time, and you were inferring shallow versus deep sources. Further back.
These are the only answers.
Yeah, keep going. Oh, are we already going?
Yeah.
Okay. Maybe can you step forward one more time? Okay.
These are the precip soil.
Maybe it's just I guess I would, this is xylem water.
Yeah,
Okay.
this is xylem water.
And you were saying something about how throughout the year, the trees, like a single tree, is either sourcing a deep or a shallow source of water.
Mm-hmm.
And I was confused by the timing because I imagine that these negative values, which we find in, especially in like the hollows, in July, which suggests a deep source, that would be the time when we're getting the most rain, the most precipitation is rain. And so why are they not tapping into whatever the isotopic value is,
that rain is
available water?
Yep, exactly.
Rather than the deep water.
Well, so I think what's happening here is, I think it's this one that you're talking about right here, is that the soil moisture data, the other site, we actually had this really strong dry down from June to July. And so this particular site where this tree grows, it's really dry. You go out there in July, it is crisp. And so I think what's happening is as the soil is dry, those other upper fine roots just die. There's no, I mean, it's so dry up there that they die. They're done for the year. And you've got the deeper roots drawing water from deeper down. So even when it rains, you know, there's not, I mean, there's this other factor of litter. So if you walk through the ponderosa pine forest, there's a lot of litter. So how much water is actually getting into the soil, you also don't know. But if they're I mean, it's pretty consistent. These are multiple trees that we're collecting from across, you know, from hollow and slope. And so they're using a deep water source. Again, this is 1 year of data, and this was a very wet year followed by a relatively dry summer. So we have 2 more years of data that we're analyzing.
So if this system is getting 50% or 56% of its water from snow, when is most of the rain delivered? I guess I would have.
Yeah, in the summertime. Yeah, June and July. It is.
So that's when it's still really
Right, but the upper elevation are using it still because the soils don't dry down as much. So the soils remain moist. I think those fine roots are still active, and then it rains, and they're using it up. Here, it's too dry, I think.
But in June and July, in the high level, they're using all of it's all showing a deep water source.
Well, yeah, so what's coming in here could be deep water or a lot more negative rain that's coming in here, too. That's why it's hard to separate that out. Yeah. Yeah, isotopes are magical. You can't tell us everything, but we're trying to untangle this a little bit.
Well, thank you very much.
Okay, thank you.